Good evening. I'm Megan Donaldson, CEO of the Hereditary Disease Foundation, and I'm delighted to welcome you to our 2020 Gala and Symposium. Thank you for joining us virtually tonight as we celebrate the promise of Huntington's disease research. Our annual event has traditionally been the time we present stunning scientific findings, and also the time we renew personal connections that so distinguish this foundation. While we are hosting the Gala virtually this year, we assure each of you that tonight we are once again bringing together our HD families, scientists, and other close friends, our Hereditary Disease Foundation family, with the spirit of passion and connectedness which have been the hallmarks of the HDF. One upside of this virtual format is that it enables us to share the evening with people from all over the world. And I would like to extend a special welcome to those of you who are attending our gala or becoming acquainted with the Hereditary Disease Foundation for the first time. Tonight, we will hear from distinguished speakers who will talk about the incredible advances in Huntington's disease research and provide updates about several promising clinical trials. We will also hear from and celebrate the successes of the winners of this year's Leslie Gary Brenner Prize for Innovation in Science, and the first annual Nancy Wexler Young Investigator Prize. Over the last few years, we have significantly increased our research funding, and we will hear about the exciting advancements that your financial commitment has made possible. So in a year when independent scientific research funding has never been more critical, please join me in renewing or increasing your financial commitment to the Hereditary Disease Foundation. Thank you for your support. Now, let's get started with the festivities. Our first speaker is a renowned scientist, a key member of the gene hunters who discovered the Huntington's disease gene, and the visionary leader of the Human Genome Project, which completely mapped and sequenced human DNA. He is the current director of the National Institutes of Health. Please join me in welcoming Dr. Francis Collins.
Good evening, everyone. It's a pleasure to join you in this gala evening, even though I wish it was in person instead of virtually, but this is what we do in the era of COVID-19. But I didn't want to miss the chance to say a few words as part of this celebration of the remarkable progress in Huntington's disease research that has been nurtured for many decades now by the Hereditary Disease Foundation. I had a personal opportunity to experience that. In fact, I think I have to say that as I have spent 100 hours a week since about February working on COVID-19, a lot of the principles of how to bring groups together and how to try to recruit the best science and put it to work are things that I saw modeled in the Hereditary Disease Foundation workshops way back when I was first invited to join that group and watched over by Milton Wexler and by his remarkable daughter, Nancy, who we are also celebrating here this evening. That Hereditary Disease Foundation gene hunting group, which invented a lot of the technologies that ultimately made the Genome Project possible, was a very different model. At that point, team science was not so much done in biology, maybe in a few other places, but this was an experiment that required a bunch of dreamers and also somebody who was going to make sure that it actually happened and we didn't all fall apart in the middle. And Nancy was the right person to inspire us, to guide us, to encourage us, to take this gaggle of 7 labs and figure out how they could become greater than the sum of the parts. And that's very much what we are all trying to do now across the board, including in COVID-19. And many of those lessons I learned because of Nancy and because of those workshops and that remarkable gene hunting group that ultimately found the gene in 1993 altogether. I remember those workshops, though, as really remarkable experiences, because people came with no plans generally of things they were going to show. Slides were generally not really approved of. You needed to describe and write on the whiteboard. And you need to talk about data that you had from last week, not something that you did a year ago that was about to get published or maybe already had been. There was a real ethic there about sharing all the things that were on your mind and brainstorming together. And that's what made it work. The workshops also had some other nice features because of Milton's connection in the Hollywood realm. We, after an exhausting Saturday of hard work on science, would get to go to one of those amazing evening parties put on in some fancy mansion, perhaps, in Santa Monica or further north. And, just hobnob with Carol Burnett or Julie Andrews or Walter Matthau. Or I would occasionally have the opportunity to bring my 2 teenage daughters for those, and they will never forget that. My younger daughter still remembers the time she almost walked in on Jack Lemmon in the bathroom. She said it was really close. It was almost an embarrassing moment that would have made news, but it didn't quite turn out that way. Anyway, those were experiences. And I got to say, the Human Genome Project built upon that team effort and a lot of those same technologies that ultimately let it be possible to read out all 3 billion letters of the human genome in 2003. And of course, Nancy was a big part of that as well, helping us think about those ethical, legal, and social issues, and ultimately getting us to the place where a bill was passed, the GINA bill, to say that genetic information may not be used against you. No, not in employment and not in healthcare. And obviously we were thinking about Huntington's disease in a lot of that deliberative process. So here we are now. My goodness, I have watched and cheered for what's happening in Huntington's disease research ever since. I'm glad to say that NIH is a part of that, currently funding about 110 projects to the tune of about $48 million. Research that starts in the very basic science arena, goes through ideas about neuroscience consequences, translational efforts, and clinical trials. And I know tonight you're going to hear a summary of where we are with the clinical trials, which is truly exciting and something I don't think I could have imagined 10 or 15 years ago, to see this proliferation of opportunities. And one can't help but be inspired and excited about some of what is now happening. I'm particularly thinking about the antisense oligonucleotide trial, where those are injected intrathecally, and we're seeing a drop in the amount of the Huntington protein, and obviously waiting to see what happens with the larger trial, looking at clinical outcomes. But that is just amazing to contemplate that we have gotten to that place. And NIH is glad to be a partner with you, but HDF in all that you have done have always been a bunch of visionaries, and particularly that's true for Nancy. Also, you're going to hear tonight about the winners of 2 prizes that I understand are being awarded: the Leslie Gary Brenner Prize for Innovation in Science, and the debut appearance of the Nancy S. Wexler Young Investigator Prize, established to honor Nancy's lifelong efforts to bring young researchers into the Huntington's research field, because that's what we need more of. And I'm delighted to see that that is getting underway. So Nancy, you and the Hereditary Disease Foundation, you exemplify all of the things that make me optimistic about where we're going, whether it's for genetic diseases, whether it's for infectious diseases like COVID. It's bringing together the people with the talent, the dream, the determination, and figuring out how to bring the best science to bear on these really hard problems. And it is pretty breathtaking to see the progress that is being made, but we still have a long way to go. And just to finish, I was thinking about how I first learned about Huntington's disease, and Nancy knows this. It was because I grew up listening to Woody Guthrie. My father had been a folk song collector, and quite literally, when I was a kid, maybe age 5 or 6, and it was time to call it a day, my parents would regularly set a 78 record in this turntable in this cramped little bedroom that I shared with my brother. And we would listen, well, to a lot of interesting things, but we would listen to a lot of Woody Guthrie. And finding out that Woody was afflicted with Huntington's disease later got my attention as I was thinking about going into medicine. And then having the occasion of encountering Nancy and knowing about this research effort, I was totally hooked. It does make me think about that Woody Guthrie song, "This Land Is Your Land." And there's a line there that I keep thinking about that's just like so perfect for this, but he couldn't have possibly known why. And that was that first line of one of the verses, "As I was walking that ribbon of highway." Well, gosh, that sounds like DNA to me. That ribbon of highway, a highway that's leading somewhere. We're all on that highway. We're all looking to get to a place where suffering from terrible diseases will be dealt with, will be eliminated. All of you, walk on that highway tonight with Nancy. Walk towards a brighter time where we figure out how to deal with Huntington's disease in a fashion that it becomes more of a topic for the history books and less of something that people today have to struggle with. That's our goal, that's our dream, and I know you share that with me. So thank you, everybody. Have a wonderful evening and a wonderful gala.
The data is fantastic. It is so optimistic and so promising.
We just programmed the right order of the A's, C's, T's, and G's that we put together to make the drug that we want. I have an example of what the finished product looks like. It's just a little white powder like this.
Huntington's. And when I held that magical vial, of course I'd like to just drink it myself, you know. Why not? You just happen to have that in your pocket. That's my disease there. You're carrying it. And another person that I'm very, very proud of is Osama, because Osama studies a very special brain cell in your brain called astrocytes. And these astrocytes play a super important role in your brain, and your brain talking to each other, brain cells communicating. And with Osama's work, he's looking, some of the astrocytes make you sick faster, some astrocytes protect you, you know. So they're, you know, have a protective factor. So what Osama's work is to actually try to look at the astrocytes and say which ones are protective and which ones make you sick faster. And Osama is winning the Nancy Wexler Award, this time the very first award we've ever given. And it's very super exciting because, you know, I think his work also is going to be incredibly important.
Hi, my name is Usama Abdelahma. I'm an instructor in neuropathology at Columbia University Irving Medical Center, and I would like to thank you for giving me the chance to tell you about my work on single nucleus RNA sequencing of the Huntington disease brain, astrocytes in the spotlight. Some background. Huntington disease, as you know, is caused by a mutation in the Huntington gene, which leads to an abnormal protein that aggregates in the cells and disturbs cellular functions. Although all brain cells carry the mutation, the disease is characteristically most severe in neurons, the processing units of our brains, causing neuronal death and loss of neuronal function. But the mutation also affects astrocytes, which are the major supportive brain cells that help maintain proper neuronal function. An example of an astrocyte is shown in the figure on the right, in which this astrocyte cytoplasm is shown in green, and this allows us to visualize the numerous extensions that extend out of the astrocyte cell body to support and ensheath neuronal connections, wrap around neuronal connections, and maintain neuronal health. In Huntington disease, astrocytes may lose their ability to support neurons. But the pathology in Huntington disease is not the same across the brain. And this is illustrated in the figure here on the right, where I'm showing you slices from the brains from people who donated their brains to scientific research in the New York Brain Bank. On the left is a case of Huntington's disease, and on the right is a case of an individual who died from other causes. And you can see that there is significant neuronal damage or significant damage to the tissue, but it's heterogeneous. So if you look at the area highlighted in yellow, which is called the caudate nucleus, there is severe shrinkage and damage to the caudate nucleus in Huntington's disease. But the cortex, which is highlighted here in orange, is less severely affected. The hypothesis I set out to address is as follows. Astrocytes in the cerebral cortex are better able to protect neurons compared to astrocytes in the caudate nucleus. This is based on the differential susceptibility to neuronal damage in the cortex and in the caudate nucleus. And the approach I took to address this question is as follows. I went to the New York Brain Bank, where we have dozens of samples that are carefully curated from donors who donated their brains to scientific research. So we have frozen brain samples from the cingulate cortex, which is less severely affected, and the caudate nucleus, which is more severely affected. I carefully dissected that tissue and extracted nuclei, which are shown here in blue. The nuclei contain DNA, which carries the genetic material and the genes. And the genes carry the instructions that can be transcribed into RNA. So RNA transports instructions from the genes to make proteins and perform cellular functions. If we can read this RNA, then we can measure gene expression, and we can learn more about the cellular functions. And the approach I took to measure this gene expression is called single nucleus RNA sequencing, whereby I measure gene expression in thousands of cells from multiple individuals by reading this RNA from the nuclei of these cells. And the results have been published in this paper I'm showing you here. And the results look like this. This is a T-distributed stochastic neighbor embedding plot in which every cell is shown here as a circle. Blue are Huntington disease cells, and red are control cells. And I'm showing you here astrocytes only. So every circle is an astrocyte. The distribution of these cells in this map is driven by differences in gene expression. You can see that Huntington disease astrocytes cluster separately and aggregate together compared to control astrocytes. And this is caused by differences in gene expression, which can be illustrated in this graph here on the right, which is called a gene expression heat map. In this heat map, every gene is shown in a row and every cell is shown in a column. Red and yellow represent high gene expression. Blue represents low gene expression, lower levels of gene expression. So you can see that there are significant differences between astrocytes of the Huntington disease, which are shown on the left, compared to astrocytes of the control condition. And some of these gene expression changes are driven by differences in expression of metallothionein genes. There are multiple of them that are increased in Huntington disease astrocytes. These proteins perform several functions that may protect neurons against damage by sequestering heavy metals and preventing oxidative stress. We see these proteins being upregulated and increased in cortical astrocytes, but we do not see the same patterns in the astrocytes in the caudate nucleus, where neurodegeneration and neuronal loss is more severe. In conclusion, I showed you that in the cortex, where neurodegeneration is less severe, astrocytes increase the expression of metallothionein proteins, which have neuroprotective potentials. Conversely, in the caudate nucleus, where neurodegeneration is more severe, astrocytes do not increase metallothioneins. In ongoing work, I'm performing experiments to confirm the neuroprotective features of metallothioneins in animal models of Huntington disease. and to leverage the neuroprotective features of cortical astrocytes to protect the more vulnerable caudate neurons. And with that, I would like to thank the Hereditary Disease Foundation and the Huntington Disease Society of America for their continuous support and funding. And in particular, I would like to thank Dr. Nancy Wexler, who's been an inspiration for me and provided me with continuous support, and to everyone else who contributed to this work. Thank you very much.
The Leslie Gary Brenner Prize for Innovation in Science was created to honor the memory of the late daughter of Frank Gehry, the foundation's founding director. The prize is awarded each year to recognize unusually innovative HD research that leads to new advances in the development of a cure. I am honored to introduce today Ai Yamamoto from Columbia University as the winner of the 2020 Leslie Gary Brenner Prize for Innovation in Science. I have followed Ai's work since she was a young graduate student in Renee Hen's lab at Columbia University, where she pursued a bold but risky thesis project in Huntington's disease. Using an elegant genetic approach, she and her colleagues were able to silence completely and at any time of their choosing the expression of a toxic fragment of abnormal Huntington in a mouse model. She found that eliminating the expression of the abnormal Huntington fragment could halt and even reverse disease in the mice, even after symptoms and neuropathology in the mice had become apparent. And this included the clearance of abnormal Huntington inclusions that had accumulated in the brains of her mice. Ai's discoveries were very important because they showed that HD could be reversed. It is not hyperbole to suggest that Ai's findings attracted many new investigators to the field and helped to spur the development of Huntington-lowering therapeutics over the past 20 years. After obtaining her PhD, Ai continued her scientific training with the Nobel Prize-winning cell biologist James Rothman, where she began to investigate a potential mechanism that could explain how the symptoms in the HD mice were reversed. She concentrated on a cellular garbage disposal process called autophagy or self-eating. Later, in her own laboratory at Columbia University, I found that one of the components of the autophagy pathway, called alpha, was essential for the cell's ability to clear abnormal Huntington inclusions. Importantly, at the same time I was studying alpha's functions in autophagy, geneticists discovered that alpha is a potent modifier gene of the Huntington's disease age of onset in the Venezuelan kindred. For example, those with the alpha variant gene can have their symptomatic onset postponed over 20 years in comparison to those without the variant gene. Ai's laboratory is now investigating in detail how the alpha variant affects HD pathogenesis, and her discoveries will undoubtedly spur the development of new therapeutics. Ai has inspired countless investigators in the Huntington's disease field to think outside of the box, and now it is my honor to present the 2020 Leslie Gary Brenner Prize for Innovation in Science to Ai Yamamoto.
So thank you very much, Scott. The receipt of this award is really quite overwhelming. I'm very honored to receive it. I would like to thank several people, if I may. So to begin, I would like to thank the Hereditary Disease Foundation. It is a foundation of families, and to have been a member of this family for so very long has been an honor indeed. Thank you. I would like to thank the awards committee. This is a committee full of scientists that I have admired throughout my career, that have continued to push me and to inspire me. And to be recognized amongst them is really quite humbling. I would like to thank the Wexler family, the brilliant vision of Milton Wexler, the steady and subdued strength of Alice Wexler, and the bubbling over exuberance of Nancy Wexler, and all those hugs and kisses over the years. Thank you, Alice, Nancy, I love you very much. Thank you. I would like to thank the Huntington's disease patients and their families. Thank you very much for your support, both with your generosity and spirit and your knowledge. And thank you so much for entrusting scientists like me with your hope. It is motivation every day. I would like to thank members of my team, as well as my past mentors, especially Jose Lucas, Renee Hen, for entrusting me with the reversible mouse model. Thank you very much also for always allowing me to take center stage. Anne Simonson for the discovery of Alfie, without whom none of this is possible. Members of my group, all of whom are brilliant in their own ways. I would like to especially thank Leora Fox, who got the Alfie work started in vivo. This was really quite courageous at the time. And Catherine Croce, who has continued to keep the flame burning brightly, even despite a global pandemic. And of course, for my last thanks, I would like to thank Frank and Berta Gary. I saved this for last largely because the words are very difficult to come by. But I would like to just say it like this. So almost 6 years ago, I had my first and only child. He is my everything. And so to receive an award to celebrate the life of your Leslie is something that I will cherish forever. Thank you very much. So, can we undo the course of Huntington's disease? So this is a question I've been asking myself now for my entire scientific career, going on 20 plus years. And, you know, we're at a point in time now where we actually might be able to. And so the answer is maybe. So how do we go about answering a question like this? We can do this by looking inside the cells. So here what you have is a picture of a brain cell, a neuron. This picture was taken by Alice Spada. And let's pretend that this neuron is inside the brain of a Huntington's disease patient. And we can kind of zoom in and kind of look inside to see what's going on. Now, one of the things that we know that's going on in the cells of a Huntington's disease patient brain is that they have two forms of instruction. One, to make Huntington, HTT. This protein is an essential protein for life. And then, due to a bit of a mistake here, known as a genetic mutation, this Huntington protein is now formed in a rogue form, a form that starts doing things the Huntington normally doesn't do, such as accumulate and aggregate and make little clumps inside of the cell. Now, whatever it is that this rogue form of Huntington is doing, it's poisonous, and it ultimately leads to the demise of the cell. Now, the poisonous action of this rogue form of Huntington is so strong that it can be captured in the humble little mouse. And Gillian Bates, in a landmark study, was able to show this. She made a mouse that made poisonous Huntington. And if you express poisonous Huntington over time, and you watch this mouse, over time, the mice start displaying symptoms that recapitulate Huntington's disease. And over time, the mouse gets sicker and sicker and sicker. Now, as a graduate student in the laboratory of Renee Hen, together with a postdoc, Jose Lucas, we wanted to understand the relationship between poisonous Huntington and sickness. That is to say, that does poisonous Huntington have to be around all the time for the mouse to stay sick? If we get rid of poisonous Huntington, can the mice get better? So to ask this question, we also made a mouse. But this time, we made a mouse much like Dr. Bates's mouse, but with a little switch that turns the thing on and off. So when the switch is on, the mouse makes poisonous Huntington, and the animal gets sicker and sicker and sicker. But then at some point, when the mouse is sick, we just said, tick, and turned the switch off, eliminating the creation of poisonous Huntington. And lo and behold, the mouse got better. So what happened? So if we go back into the cells, in essence, what would happen is that we got rid of the creation of this rogue form of Huntington. And by doing so, we were able to prevent it from doing these things that it does that wreaks havoc inside the cell, like make these little clumps. Now, This is really an exciting time because, of course, the ASO trials are actually recapitulating this in patients. And so I, too, am awaiting anxiously for the results. And you might say, okay, so we've solved this question. But I would argue no. And the reason for that is, well, there's another question that we really need to answer. And that has to do with these clumps. Now, let me tell you something about these clumps. These things don't form in a healthy brain. And while they form in the brain for a patient with Huntington's disease, they also form in the brains of Alzheimer's disease, Parkinson's disease, and the spinal cord of a patient with ALS. And we were wanting to understand, are these clumps what's poisonous? Is this what's wreaking havoc to the cells? So to answer this question, what we wanted to do was try to get a sense of whether or not we can ask the cell to start getting rid of these clumps. Is there such a way? And guess what? There is. And it's in the form of this guy. So this guy's, of course, Pac-Man. He runs around in like a dark maze, eating up these little pellets. And meanwhile, he's getting chased by ghosts that want to kill him. Now, the goal is to eat these power pellets, and this is what makes Pac-Man invincible and kills the ghosts. Well, in every single cell of your body, there is a Pac-Man, and it's known as autophagy. And this pathway, autophagy, can eat aggregates. Now, you might be thinking, well, if there's a pathway, autophagy, or this Pac-Man dude that can go around and eat aggregates, why do they even ever form inside the brain? Well, the answer, we think, is something like this. So to Pac-Man, the cells look like this. There are all sorts of things it wants to eat. So what we need to do is try to think of a way to make Pac-Man prioritize those clumps. Eat up, say, chocolate macaroons before anything else. So how can we go about doing that? Well, that took a while for us to try to figure out. But lo and behold, we think we have. And it's all about this guy, Alpha. So Alpha is a protein that can label these proteinaceous clumps inside the cell. And by labeling these clumps, it can convince Pac-Man to eat them first. And so we figured if we can take advantage of this. So back inside the cell, maybe what we can do is put Alpha in them. Alpha will then label the clumps and Pac-Man can go about eating them. And then we can find out whether or not this can bring about any sort of benefit. So Catherine Kircha in the lab is currently doing just that. And so what she created was not just a mouse that makes poisonous Huntington, but she made a super mouse that not only makes poisonous Huntington, but also makes a lot of Alpha. And what we're doing now is looking to see what will happen to those symptoms that look like Huntington's disease. Now, we're still in very early phases of this study, and so we're not quite done. But what I can report is something very exciting, and it's the following. What Catherine is seeing is that the mice just aren't getting sick, or not as fast anyway. And she's so excited that now she's asking that next question. If we take a mouse that's sick, with the cells full of these clumps, and now we put in Alpha into those cells, do the mice get better? I unfortunately don't have the answer to that question right now. But before I go, I just want to thank a few people that helped along the way. This is Jose Lucas and Renee Hen, with whom we created the mouse with the little switch. This is Ann Simonson. She's the person that discovered Alpha. My lab members, current and past, the ones in yellow are the ones that directly contributed to this work. And of course, this work was utterly impossible without the support from you, as well as the Columbia University, my home institution, and ever importantly, the NIH. Thank you very much.
Good evening and welcome to the 2020 HDF Gala. I'm Ann Young from Massachusetts General Hospital, and I am chair of the Science Advisory Committee of the HDF. And tonight, you have already heard very exciting things that are happening, but I wanted to introduce people who are going to tell us about clinical trials for actual therapies for Huntington's disease that make a difference. And next, we're going to have David Cooper from Unicor, and he's going to be telling us about their genetic technologies for therapy in Huntington's disease.
Hello, my name is David Cooper, and I'm the Vice President of Clinical Development, responsible for the HD program here at Unicure. I'm honored to introduce our company and our gene therapy clinical development program to all of you. I'd like to say thank you to the amazing team at the HDF Gala for giving us this opportunity to present an update on our program. It's been a very exciting year as we've moved this trial from the startup phase through treatment of the first 4 patients. Unicure is a gene therapy company dedicated to developing AAV-based treatments for patients with high unmet medical need. For those diseases where there is a genetic link, gene therapy represents a very promising treatment approach. Gene therapy is defined as a technique that uses genetic material to treat or prevent disease. In the case of CNS or brain diseases, if the gene therapy package is delivered to the brain itself can be the factory that reads the instructions to produce the therapy to correct the genetic defect. Unicure is developing a gene therapy approach to reduce the amount of mutant Huntington protein that's produced, which is the cause of HD. The gene therapy is based on an adeno-associated virus, or AAV5 capsid, that contains DNA instructions to make a microRNA against the Huntington RNA, or my-HTT. We call this AMT-130. The clinical trial we're conducting is the first gene therapy to enter clinical testing for the treatment of HD. AMT-130 is administered only once by very targeted neurosurgical procedure that uses intraoperative MRI to target the relevant structures and monitor delivery of treatment. There are 2 key components of AMT-130, an AAV5 vector and a gene encoding the microRNA. The vector acts as the delivery system. It's based on a non-disease causing AAV5. It's been modified to deliver a gene encoding a microRNA that will recognize, bind, and then non-selectively lower human Huntington protein. The main purpose of the study is to find a safe dose of AMT-130 in adults who have tested positive for the HD gene and who have early stage HD. The study will also look at how the body processes AMT-130 and will explore how AMT-130 might affect how the disease progresses. Ultimately, what we'd like to learn is if AMT-130 can lower Huntington protein levels and thereby slow the progression of Huntington's disease. The objectives of the phase 1/2 study are to assess the safety, tolerability, and efficacy of AMT-130. We're using a double-blinded approach comparing AMT-130 at 2 dose levels to imitation or sham surgery. Double-blinded means that neither the patient, the HD neurologist investigator, or the blinded study staff will know if the patient is in the treated group or the imitation group. As I only have 5 to 6 minutes, I can't go into the details about eligibility, and the most up-to-date information is always posted on clinicaltrials.gov. Eligible patients will have a genetically confirmed CAG expansion in the Huntington gene and early manifest or diagnosed HD. As we've learned from screening patients this year in our trial, we have recently updated these inclusion-exclusion criteria. I wanted to highlight that the population of early manifest HD is now broadened to include those with either predominantly motor symptoms of HD, so-called motor manifest HD, and those who have more of a combination of motor, cognitive, and behavioral symptoms, termed multidimensional manifest HD. The study is testing 2 dosing levels of AMT-130, with the safety of the low dose being assessed before we begin testing the high dose. Procedures will include clinic visits, assessments of physical and neurological health, a neurosurgical procedure, lumbar punctures, brain scans, and samples of other bodily fluids. The blinded part of the study has frequent visits to assess safety, efficacy, and biomarkers and lasts for 12 months. Thereafter, periodic visits will continue through 5 years of follow-up for those patients who received AMT-130. Following preliminary safety data and efficacy data review by our Data Safety Monitoring Board and the FDA, Patients who were assigned to the imitation surgery group who complete the 12-month follow-up and still meet eligibility criteria may be able to receive AMT-130. Here are some recent updates to the trial. We're happy to announce that the first 2 patients were dosed in June of 2020, and the second 2 patients were treated in this past October. Overall, it's been a very busy year for us and a very productive one for moving the trial forward. Here is the list of the 9 sites that are currently recruiting patients in the United States. The trial site list is continuously updated on clinicaltrials.gov. We believe that the best way to understand the impact of HD is by engaging with the community. So we work to create opportunities for the patient voice to be heard directly via patient speakers internally and patient advisory boards. We use that knowledge to help shape internal programs and identify opportunities to support the community. On behalf of the uniQure HD team, we would like to again thank HDF for the opportunity to present an update on our HD clinical program. For more information about the program, you can go online or email us.
And next, we're going to have Paul Bono from Wave Pharmaceuticals. They have a very interesting genetic lowering fat therapy, and they will be presenting their progress in that so far.
Good evening. My name is Paul Bono. I am president and CEO of Wave Life Sciences, a genetic medicines company based in Cambridge, Massachusetts. Thank you to all those tuning in. I hope you and your families are staying healthy and safe during these challenging times. And thanks very much to HDF for this opportunity. We've been so grateful for your partnership over the years, and we're inspired by the leadership that you provide in supporting and funding innovative research to treat and eventually cure Huntington's disease. At Wave, we're focused on delivering transformational therapies for patients with serious genetically defined diseases. Wave was founded to advance an innovative and unique discovery and drug development platform focused on treating serious illnesses such as Huntington's disease. And we're excited about the progress we've been able to make with the tremendous partnership and support of the HD community. Our current clinical programs are in Huntington's disease, where we are conducting 2 phase 1b, 2a clinical trials. We expect to initiate clinical development for a 3rd HD program with the submission of a clinical trial application in the 4th quarter of 2020. What is especially exciting about all 3 of these programs is that they are allele selective. That is, we aim to selectively lower mutant Huntington protein while leaving healthy Huntington protein relatively intact. To do so, we target a site called a SNP, or a single nucleotide polymorphism on the mutant copy of the Huntington gene. We liken these SNPs to a pin on a map directing our molecules to a precise location on the mutant copy of the Huntington gene. The premise behind our innovative approach is to preserve the healthy Huntington protein, allowing it to do its important work in maintaining and protecting the brain, especially in the presence of cellular stress, as occurs with the buildup of mutant Huntington protein. Given the role that healthy Huntington protein plays in so many critical biological functions, both in the central nervous system and systemically, we believe that the potential benefit of our approach for Huntington's patients is significant and may offer promise in addressing the disease well before symptoms manifest. Our 2 ongoing clinical trials in HD are called Precision HD1 and Precision HD2, and are designed for adult patients with early manifest HD who carry SNP1 or SNP2 respectively. In December of 2019, we provided an interim update from the Precision HD2 trial that supported clinical study continuation, including the addition of a higher dose cohort, which we initiated for both our Precision HD1 and Precision HD2 trials earlier this year. While the impact of the coronavirus has been unavoidable for so many of us, our clinical trials and our work in the labs has continued to advance. Even as we remain keenly focused on ensuring the safety of our study teams, patients, and investigators, neither our commitment nor that of the investigators and the patients participating in our studies has changed because of the pandemic. We expect to announce additional clinical data in the 1st quarter of 2021 from all treatment groups for both the SNP1 and SNP2 trials, and available data from our open label extension studies, which we implemented to assess long-term safety and tolerability. We have also continued to successfully advance the work required to submit the clinical trial applications for a third HD program in the fourth quarter of this year. This third allele-selective program will target SNP3 and incorporates all of our learnings from our SNP1 and SNP2 programs, as well as the advancements in our platform, including new chemical modifications that show improved potency and durability in preclinical experiments. Importantly, targeting SNP3 increases the number of HD patients who could potentially benefit from our innovative approach. This program addresses a significant portion of the Huntington's disease population on its own, as approximately 40% of the HD population carries SNP3, an observation that we have confirmed using patient samples from our observational study. Together with our SNP1 and SNP2 programs, we have the potential to address up to 80% of the HD population with selective mutant Huntington lowering. In addition to advancing our research on candidate therapeutics in HD, we've made significant progress in evolving our methods to identify patients eligible for SNP targeting and in the measure of biomarkers to help evaluate our candidates in the clinic. Our work on these scientific challenges has the potential not only to help make our programs more efficient and position us well to achieve a clear clinical outcome, but more importantly, may help advance HD research more broadly. As we continue our work at Wave, we recognize that we could not do what we do without the broader HD community. Our goal is to ensure that the voices of patients, families, caregivers, advocacy organizations, and other members of the community drive and inform our work at every step. In our quest to deliver meaningful disease-modifying therapies, we are grateful for the support and guidance of the HD community and organizations like HDF, whose leadership have resulted in hopeful advances that we're discussing today, from improvements in our understanding of the biology of HD to advancements in genomics, testing, and treatments. In particular, we want to take this opportunity to thank all those participating in our trials, including those who have participated and continue to participate during this unprecedented pandemic, as well as those whose partnership has been critical in getting these potential therapies to this point. I'd just like to close this evening by sharing a short video to highlight our appreciation for this community. Thank you again, and we look forward to continuing to move forward together.
I'm delighted to introduce Dr. Chung Ho Cha from Novartis. I am also particularly happy because he was a graduate student with me and also was on the faculty at Mass General before he went into industry to try to find better therapies for Huntington's disease. And he will tell you tonight about some very exciting strategies that they are taking.
Good evening. My name is Chung Ho Cha. I work at Novartis, and it's my pleasure to be with you tonight. And I'm going to tell you about Ranaplam, our new Huntington-lowering compound, on behalf of my colleagues Rajiv Sivsankaran and Beth Borowski and the LMI 070 team. Ranaplam is an RNA splice modulator. So this basically alters the way that mRNA molecules are spliced. And we're testing it right now in spinal muscular atrophy, which is a severe motor neuron disease of babies. In that disease, the babies with SMA are lacking a protein called SMN, and Branoplam allows for the retention of an exon in the mRNA, which allows the SMN protein to get produced and retained. So through the action on mRNA splicing, Branoplam for SMA allows SMN protein to be made. But it turns out that Branoplam affects mRNA levels of other molecules too. And in this plot on the left, it's what's called a volcano plot. One of the molecules that is decreased is Huntington. So Branoplam, presumably through a splicing mechanism, decreases the mRNA levels of Huntington. And remember, mRNA is what makes the protein. And so if we test this on the right, we see that Branoplam actually does decrease the amount of Huntington, and whereas an inactive analog does not. And so we have some insight now into how Branoplam affects the Huntington mRNA levels. I've told you that Branoplam is a splicing modulator, and in the bottom, what we now know is that Branoplam will create the inclusions of what's called a pseudoexon in the Huntington mRNA molecule. Well, this pseudoexon, which is in pink, leads that mRNA molecule to get degraded. So you create a defective copy of the mRNA and it gets degraded so the protein cannot be produced. And we see that this is true in cell models. So on the left, we see that this novel exon, the pseudoexon, gets increased with the amount of Branoplam. Because this is a defective mRNA, it actually lowers the amount of total mRNA for Huntington. And then correspondingly in the bottom left, you see that the amount of protein also decreases. And we've seen the same things in human cells. These are fibroblast cells from human HD patients. And on the top that we see that the Branoplam will increase the amount of this poisoned mRNA, and correspondingly on the bottom right, it lowers the total amount of Huntington protein. So it's all going in the right direction. Working with Bev Davidson of Children's Hospital of Pennsylvania, who is a HDF SAB member, we tested this in back HD mice made by William Yang, another SAB member, and we see that Branoplam lowers the Huntington transcript and lowers the Huntington protein in these mice as well. And this is oral dosing. This is an oral medication given 3 times a week, and we see nice decreases in the amount of Huntington protein. So as I told you, the Branoplam is being explored now in one disease, spinal muscular atrophy, and those studies are continuing, and we're learning a lot from that clinical program. But we're also bringing it into Huntington's disease. Now, Ranaplam has never been given to adults because the kids in the SMA clinical trial are infants. So right now we're doing a phase 1 study in healthy adults to understand how Ranaplam is metabolized in adults. In 2021, we're going to start the phase 2B study in HD patients to see if Ranaplam is effective in that patient population. So we have learned a lot from the ongoing clinical trial in SMA, and to make a long story short, that continuing study tells us that the doses are well tolerated, that the adverse events so far are mild to moderate, and so far the kids seem to be doing well. So this gives us a lot of confidence to bring this molecule into the HD population. The other thing is we've been able to examine blood samples from the SMA infants. Remember, we all have Huntington mRNA, and we were able to look at what happens to the Huntington mRNA in the kids that have spinal muscular atrophy. And you can see in the top line that their levels of Huntington mRNA decreases, indicating that the Branoplam is in fact decreasing Huntington's mRNA in human patients. So we're going to start in the new year a phase 2B study in Huntington's patients. This will be a study done with early stage 1 or stage 2 Huntington's males and females. The drug will be Ranaplam, which is an oral medication. And it looks now like we'll be able to dose either weekly or at most twice weekly. And what the right dose will be informed by the ongoing SMA trial, in addition to that phase 1 trial that's ongoing currently. And we'll do both in-clinic and at-home nursing assessments. And then after a period of 16 weeks, all of the patients will get rolled over into an open label extension, so we can see what the long-term effects of this medication are. So we're excited about the possibilities and the possible benefits of using an oral medication. First of all, it's going to be a lot easier for patients to take an oral medication. We also think that an oral medication, because it's distributed throughout the body, has a better chance of reducing Huntington not only in the cortex, but in the deeper parts of the brain. We know that the striatum is the area that has the most damage from Huntington's. And finally, because Huntington is expressed throughout the body, there's the possibility that an oral medication might have benefit from lowering Huntington in those peripheral tissues. So I speak on behalf of a large, very motivated team, and we're tremendously excited, and especially with the announcement last month from the FDA that Branoplam has received orphan drug designation. So we are working as hard as we can, and I would just like to also give a shout out to two of my mentors, Ann Young and Nancy Wexler. Thank you for your attention.
Sarah Tabrisi from the University of London will tell us about the ongoing treatment of Huntington's patients with an antisense oligonucleotide to lower the protein. And it is now in phase 3 clinical trials with Roche and Ionis.
Thank you for inviting me to give you an update from the Tolerability Global Clinical Development Program. My disclosures: in the past 2 years through UCL Consulting, I've provided consultancy services for Roche and Genentech. I'd like to start by thanking all of the global investigators working on the Tolerability Clinical Development Program, and to express my deepest gratitude to all past, present, and future study participants, as well as their families. There are 4 objectives in my presentation. The first, to provide a high-level overview of tominersen and the clinical development program. 2, to share the latest insights from the open-label extension of the phase 1/2a study. 3, to provide a brief overview of Generation HD1 study status. And 4, to discuss the impact of COVID-19 on the tominersen clinical development program. Tominersen is designed to lower the production of the toxic mutant Huntington protein. The clinical development program consists of 5 studies which aim to answer 3 key questions. What is the optimal dosing strategy for efficacy, safety, and tolerability? Do any safety concerns emerge when we treat a larger group of people for a longer time? And does sustained treatment slow or stop the progression of Huntington's disease? The tominersen clinical development program is generating data to evaluate the efficacy and safety of tominersen. The original phase 1/2a study is complete and was published last year in the New England Journal of Medicine, and Nancy Wexler and Kurt Fischbeck did a wonderful editorial in the same issue. All of those subjects rolled over into the open label extension, which is complete and final analysis ongoing. The Huntington's disease natural history study is ongoing and recruitment is complete. And Generation HD1 is the phase 3 study. This is ongoing and recruitment is complete. The latest insights from the open label extension. These graphs show sustained lowering of CSF mutant huntingtin, our target, and this was observed in both treatment arms at 15 months. All of the individuals in the original phase 1/2 study were randomized to receive tominersen every 4 weeks, shown on the left, or tominersen every 8 weeks, shown on the right. On the y-axis is CSF mutant huntingtin change from baseline, and on the x-axis is visit day. You can see here there's a 70% mean trough lowering at 15 months in the 4-weekly cohort and 44% mean trough lowering at 15 months in the 8-weekly cohort. The target range is 30 to 50% lowering. Pharmacologically relevant CSF mutant huntingtin lowering was observed in both treatment arms, but the data shows from the open label extension that 8-weekly dosing is sufficient to reach our target CSF mutant huntingtin reduction. This is a busy slide, but it shows the safety and tolerability data. And the tominersen 8-weekly at 120 milligrams appears to be more suitable for chronic dosing than the 4-weekly based on the tolerability and safety data. I want you to focus on the red boxes here. In the 8-weekly dosed arm, there were 218 adverse events. Only 2% were considered drug-related. There were no drug-related serious adverse events, and there were no SUSARs. There were no ASO class risks associated with either regime, 4-weekly or 8-weekly tominersen. So from the open label extension summary, pharmacologically relevant CSF mutant huntingtin lowering was observed. 8-weekly dosing was sufficient to reach target CSF mutant huntingtin reduction. The 8-weekly regime is better tolerated than the 4-weekly regime in terms of number and nature of adverse events and serious adverse events. There was no evidence of ASO-associated class risks in either regime. And based on the data from the open label extension, we've moved forward with an 8-weekly dosing regime in Generation HD1. Generation HD1 is the phase 3 study. I'm pleased to tell you that recruitment is now complete. 791 subjects with manifest Huntington's disease have been recruited at about 100 study sites in 18 countries. This is a randomized, multi-center, double-blind, placebo-controlled study. It's comparing tominersen 120 milligrams every 8 weeks to tominersen 120 milligrams every 16 weeks compared to placebo for 25 months. And it will evaluate the efficacy and safety of intrathecally administered tominersen in adult patients with manifest Huntington's disease. It's a properly powered study that is designed to give us the answer of whether regular tominersen slows progression of Huntington's disease. Impact of COVID. COVID-19 has been an unprecedented challenge for all clinical trials programs worldwide. It's been an unprecedented challenge for all of us. Roche is actively managing the situation by respecting local laws and guidelines, having close contact with trial stakeholders, rigorously recording patient data, and formulating mitigation strategies. Fortunately, Generation HD1 is a particularly robust study due to its large sample size and geographic diversity. And importantly also, missed doses do not mean discontinuation from the study. So in summary, the open label extension has confirmed that 8-weekly dosing was safer and better tolerated than 4-weekly. Generation HD1 study, the phase 3, is now fully recruited with 791 participants. And we hope to get the readout for this in 2022. Roche is actively managing the COVID-19 situation, formulating strategies to mitigate any effects it may have on their ongoing trials. And finally, another thank you. A big thank you to all the global investigators working on the tominersen Global Clinical Development Program, to all past, present, and future study participants, as well as their families, and your ongoing contributions to the program during these uncertain times are inspirational. Thank you for listening.
Hi, everyone. It's really a pleasure to welcome you to tonight's Q&A session for the HDF Gala. And first of all, I'd like to congratulate both Osama and I, Yamamoto. They're here part of our panel. So we have all the speakers from tonight. We have Osama and I, Yamamoto, and we have David Q. Cooper from Uniquear, Paul Bono from Wave, Chung Ho Cha from Novartis, and stand-in for Sarah. So since it's the middle of the night there in London, Dr. Blair Levitt is graciously joining us from University of British Columbia. He's the director of the Center for HD Research there and an investigator for the Roche trial. So welcome everyone, and thank you so much for being part of the panel. And so I'll be getting questions from the Q&A box and passing them along to you. And we'll start out with Osama. So this is from Christopher Pearson saying, thank you, Osama, for the fantastic presentation. Congratulations on winning this highly prestigious award with the namesake of Nancy Wexler, a leader in HD research herself. And he's asking if you analyzed or assessed the degree of somatic CAG size and heterogeneity.
Well, thank you, Leslie, and thank you, Chris, for the question. We're doing that. We're measuring CAG repeats from different brain regions. We haven't yet found out a way to measure CAG repeat expansion in single cells, but it's something on our radar. But as of now, we have the data on some samples, but not all the samples.
Okay. And okay, and Blair, maybe just there's, I'll go ahead and ask a question. In your experience, so Sarah was talking about the complications of COVID and in her center. I just wondered what your experience is and sort of how you've been interacting with the families and the patients.
Challenging time for everyone. And for the most part, we've had to convert to almost exclusively virtual visits for our patients. But obviously that can't be done with an investigational therapy that needs to be administered intrathecally. So we've been able to actually do very well. And I think we only missed one dosing at our site. We were able to get exemptions due to the requirement for the intrathecal administration. So I think most of the sites, we've been successful in carrying forward and continuing the trial with very little interruption. And so, yeah, it's been challenging. We've had to modify all of our protocols, like I think everyone has in their clinics, with extensive, requirements for cleanliness. But we've been very fortunate to work with our local IRB and our local clinical site to be able to keep the trial going. Okay,
thanks very much. Also, another question, great presentation, Chung Ho. In the necessarily long-term treatment of SMA or HD individuals with Branoplam, what might the off-target side effects be?
Well, we're trying to understand that. Of course, Huntington is expressed everywhere in the body, and with an orally delivered drug, we would expect that there would be Huntington lowering in other parts of the body besides the brain. This might have some beneficial effects. People have really considered whether mutant Huntington in tissues like the heart or in skeletal muscle or even in the pancreas might be contributing to some of the symptoms you see in the HD population. But also, there might be a an unknown deleterious effect to lowering Huntington there. We think that's one of the advantages of an oral medication, that if there are adverse side effects that we can see, then we can stop the medication, and perhaps some of those side effects are reversible, and then you can give somebody a break. But we'd have to see, and this is why these trials are so important, so we can understand exactly what happens.
Thanks very much. And I'll just ask the people that are writing questions in the Q&A box to let me know who you're directing your question to. In some cases I can tell, but not in all of them. And I guess this is also for you, Chung Ho, as maybe not, maybe for Roche. Was the decreased Huntington mRNA assessed in the CSF? Or any of you, I guess? Any of the people doing the clinical trials, have people been assessing besides Roche, I know Roche has been looking at mRNA, or it has been looking at protein. Was mRNA assessed?
So I can quickly answer. In the Roche trial, we were not looking at mRNA, we were looking at the protein levels in the CSF.
Okay, thanks. And another question for you, Blair. If approved, the thought that we won't be able to afford these medications is terrifying. And so asking for comments, and I think that's a fairly broad question.
Yeah, I think that would be a terrifying situation, and none of us anticipate that being the case. I can't comment on what the ultimate pricing will be, but certainly when this question has come up with my Roche colleagues, they've made assurances that it will be made available to all the patients that need it. But I can't comment directly on pricing.
Okay, thanks very much. This question might be directed to you. Since HD results in the death of medium spiny neurons, are there any metrics or tipping on the tipping point? So up to what point do you think symptoms might be rescued? And maybe from your autophagy background and your mouse model that you had done, and then we can get into the clinical aspect of it.
One of the caveats of working with mice is that we work with very young animals. And so, no matter how much we age them, we don't have the patience to wait until they get as old as typically what the Huntington's disease patient might be experiencing. And so, that is one of the concerns. And I think what the trials will allow us to learn is at what point can people come back. I'm forever an optimist, so I imagine that they will come back. It doesn't matter when you start these treatments. But of course, earlier the better. Something I'm learning as I'm getting older. So.
Yeah. And maybe Paul, do you have any comments on that?
Yeah, no, it's a great point, and I think all of us share your optimism, and think you have to stay in the field of research and development. I think our enthusiasm and excitement is about how do we think about moving earlier in the disease setting. And I think when we think about the opportunity, not just for looking at patients with more advanced disease and studying that impact, but really how do you move into the pre-manifest setting of the treatment of Huntington's disease before symptoms, when, as you pointed out, the possibility of reversal or the prevention of increasing detriment, you can have an impact. And it was a large driver for us in focusing on allele-specific therapies. So removing that mutant protein insult from those cells, but preserving the natural healthy Huntington protein, which is important. You mentioned it's ubiquitous in all parts of the body. So, we really think about how do you diminish that mutant protein, but preserve that healthy protein so that you can envision a future where you treat earlier at the genetic diagnosis rather than the symptomatic diagnosis.
Thank you very much. We have a question also from CERM. This is for Blair, are the adverse effects seen in the Q4W dose of tominersen related to decrease of normal Huntington protein?
An excellent question for which there's no real way to answer it. There were very few of the adverse events that were felt to be related to study drug, in fact. Most of them are related to procedures. So if you look at the adverse events overall, 4-week dosing, so an infusion every 4 weeks versus 8 weeks, actually had pretty close to twice as many adverse events, most of which are mild and self-limited. So I would say overall, no, not at all. It's related to the frequency of procedure. And one of the really great things about our data from the open label extension is it's allowed us to have confidence moving forward in the phase 3 trial to now look at Q8 week versus Q16 week. So, at one point we were looking at 12 doses in a year, and now we're looking at 3 doses in a year. And so given that the incidence of side effects is primarily related to the procedure, I think that's an incredible advance.
Thank you very much. Another question from UCSD, and I think I'll take a second to just also welcome all the people. We have an incredible number of people who registered from all over the world. We have a big contingency from Spain, from many different countries. And so this question in particular is from the UCSD, and then we have the next one is from Australia. So hi, Osama, thank you for your great presentation. How did you separate astrocytes from the other cell types like neurons, radioglial cells, or the Rna-Seq in different brain areas?
Thank you for this question. The nice thing about the technique that I'm employing and others in the field are using the same technique or variations on it is that you don't need to subject the tissue to any specific treatment to enrich for one cell type or the other. For example, in some cases when you are doing whole cell, single cell sequencing, you can enrich for neurons or microglia by using markers. When you do single nucleus RNA sequencing for, and you want to specifically enrich for neurons, you can flow sort or FACS sort these neurons using specific antigens. But if you don't do that, then you get a somewhat unbiased sampling of all the cells that you have in the tissue. And then by virtue of the uniqueness of the transcriptional programs that distinguish neurons from astrocytes, from oligodendrocytes, from microglia, then you can assign cells based, cell lineages and cell types, including astrocytes and other cell types, based on the combined gene expression. And we do have a good idea about what the gene expression of an astrocyte looks like in controlled conditions, and what a neuron looks like in controlled conditions. And oftentimes these identities are retained, but there are features that make them appear reactive or diseased in the condition, in this case in Huntington's disease. So we do have an idea from the literature and from the published work about what cell lineages and cell types look like, so we don't need to subject the tissue to any specific treatment. I hope that answers the question.
Thank you, Osama. From Australia, we have congratulations all. I had a general question regarding the increasing evidence that changes in HD start very early, perhaps in development, affect biological systems outside the brain. How might the various therapeutic strategies be adapted after positive results in symptomatic patients to earlier stages of disease and to treating the body as well as brain pathology? And maybe I'll start asking David this and then go to Chung Ho, who has the oral version. So David, do you have any comments on that?
So I think we've certainly looked at a lot of disorders that have combined CNS and peripheral features to them, and looked at what are the different approaches to do gene therapy. And from the approach we're doing currently, we're very much focused on, can we inject into striatum and get a response, and what extent of knockdown do we need to do to get that response? As we look to go more broadly and come up with next generation approaches, when you use cisterna magna, for example, and inject into the CSF, you do get some systemic recirculation. So there are other ways from a CNS injection of gene therapy to also get some CNS, some non-CNS coverage. And you can certainly do a dual injection of IV plus CNS. So I think our primary goal now is establish that it works in the CNS if you apply gene therapy right to the site of the injury and the putamen and the caudate. Secondarily, next gen would be, how do we get it systemically as well if we need to?
Thank you. And Chung Ho, with the oral application of drug, and also just this whole idea maybe about the developmental aspects of this and other tissues.
Yeah, so it's a great question, and it's something really that's fascinating to think about. And what the role of Huntington especially is going as far back as embryonic development. We know that if you don't have Huntington protein at all, you have embryonic lethality. So it's doing something very important early on. But we also see that there are early developmental effects that are quite subtle, and we don't know the import of these effects to this point. So I just picking up on Ai's comment before about reversibility, I think it makes a lot of sense to go as early as possible. Certainly once neurons, whether they're medium spiny neurons or resistant astrocytes from the cortex, once the neurons are gone, there's not really any bringing them back. So we think preservation will, an ounce of prevention is always better. Now, with the genetic diagnosis, one could conceivably treat someone as soon as they're genetically tested and start them on a therapy. But then it's kind of a balancing act, because every medication, every drug, every treatment will have benefits and risks. And those risks for medications, you wouldn't take them necessarily unless you knew that there was going to have benefit. And we just don't know that yet. So if we have efficacy in people who have clear evidence of disease, that's encouraging. And then you would want to move earlier. And then how much earlier? One could imagine a time when you start the treatment at the time of genetic diagnosis, as Paul so eloquently said. But that's going to be a balancing act of the toxicities and side effects of all of these treatments versus the demonstrated efficacy.
Thank you. And just read a comment here. My heartfelt thank you to all of you that worked so hard to help us beat this horrible disease. And they loved the Pac-Man analogy. And now a question for you, Thanks for your great talk. Do you know the mechanism by which Alfie recognizes specifically Huntington aggregates? And thanks again.
So Anne Simonson, who uncovered the protein, has done a lot of work in this regard. And so aggregates inside the brain, it's not just Huntington aggregates. Alfie can recognize other aggregates of other diseases as well, which what makes it so potent. There's a signal inside cells that actually tell the cell that you have an aggregate. They don't like having them there. And so it looks like Alfie responds to that. Now, exactly how that's happening, like down to the nitty gritty, we're still working out. But the general concept, I think Anne's work has shown is the case.
Okay, thanks very much. Here's a question, maybe open this up to the people doing the clinical trials. Treatments discussed tonight seem to be targeting very early stage HD. There's a large population of HD patients in stage 2 and 3 HD. Are any of these drug therapies going to be tested in the near term with patients whose disease progressed past early stage? And if not, what message do you have for people in that population? Kind of open this up, maybe Blair, Chung Ho, David, Paul.
I mean, I think all of us. You were on the latest side of this study, and I'm happy to start earlier, but I think the last message is probably the most important, which is hope. And I think, to be at a point where we talk about the opportunity to talk about mutant Huntington depleting strategies and therapies that are not just ideas and not just treating cells in a dish, but actually being evaluated in the clinical setting across all of these different formats, I think creates hope. I think the key that all of us have are operating in an environment where we need to translate that and see that activity where it can be measured in patients. And once we can see that and move that forward, then that opens up a host of possibilities. But I think the message is hope. I don't know, Blair.
Yeah, I couldn't have said it any better, Paul. I think we're excited to be where we are, and these trials take longer than any of us would like. But unfortunately, we have to do the hard work. And I would anticipate that a positive result in any of these studies would prompt further studies in more diverse patient groups. So absolutely, the message is hope. And yes, I think absolutely we would like to see trials in all categories of Huntington's disease.
Thank you. This is a question from MIT.
I'm happy to add to that a little. So I agree. It's a very exciting time. I think we started actually looking for sentinel patients in stage 2, and because of the unanswered questions about MRI striatal volumes, we ended up now closer to very much stage 1 and including GCL3 in our current inclusion criteria based on negotiations with regulators. But I think what happens once we actually show with any of these exciting therapies that they work is really going to be exciting. How do we modify them for even earlier patients, prodromal, and even later patients? But I think we have to figure out how to prove that they work first.
Okay.
That's the key.
Thank you very much, David. I'm going to hit you up, Chung Ho, in a second. This is just one last quick question, and then I'll close it with the final one. And this is from MIT. To all the clinical trial, is NFL light chain being measured? And if so, is it measured in the CSF or circulation? And does the trial allow use of tetrabenazine? Maybe if one or two of you might have an answer for that.
Yes, plasma, CSF.
Yeah, plasma and CSF.
Yes. We're measuring CSF, NFL, as well as mutant protein.
And people on tetrabenazine, Paul?
I do not know. That I would have to check our inclusion.
Okay. Okay, just to end up then, one of the and there's other questions, but I'll stop with this. Thanks for sharing tonight. And what is your message to the young people still at risk or pre-symptomatic and thinking about their future in regards to HD? I think we've heard a lot of comments about how much hope we have and how much hope there is now. Chung Ho, did you have any other comments?
Yeah, I just had an editorial comment about stage 1 and stage 2 patients. I don't think any of us believe that they're more meritorious than anyone who has HD, but I think there's practical reason, and these are the patients in which it's easiest to detect if the drug is having a benefit or not. So in a pre-symptomatic patient, you may not be able to tell if the drug is having any effect. But in stage 1 and stage 2, that's easy. So they're the most informative in a way, but I think anything that would work in stage 1 and stage 2 would very likely work for all patients with HD. Just that editorial comment. So I think it's a practical, and it's a way to get the trials done with the fewest number of patients and therefore the most quickly.
Thank you. Right. And then for those who are pre-symptomatic or any other comments?
I mean, beyond, I would say hope is important, but I think the excitement and understanding of biology. I mean, I think if we think about a time in biology of Huntington's disease and through, a massive effort right across universities, patient advocacy organizations, the investment in trying to really understand the biology of the disease, where now we're at a point in time where the tools are there across whether it's gene therapy, oligonucleotide, genetic medicines, small molecules. I think the ability to train on the multifaceted approaches of Huntington's disease and understanding, is a time where hopefully can become a reality beyond just wishing for something to happen. But I think all of us are excited about making something happen.
I think that's a perfect end to this session. And like to, first of all, thank you all for joining us to celebrate the promise of Huntington's disease research and the hope that it's garnered. And I'd like to raise a glass, a toast to everyone here on this panel, everyone who's participated tonight, all the researchers, to the HDF, and to our beloved Nancy Wexler. And raise a glass, and thank you so much for joining us tonight. Good night.
Good night.